Greetings to all the viewers. The session is going to cover with the compound called hemocyanin. This is the part of bioinorganic chemistry. We are treating with bioinorganic chemistry where hemocyanin is the content we are choosing for the day, right? The session is powered with AI, artificial intelligence powered session. It will be preferred IUPAC name for this bioinorganic compound will be hemocyanin, where copper taken as a transition metal in order to get the coordinate covalent complex, right? So this is the way hemocyanin is the coloring pigment in some animals. Those who are accompanied by hemocyanin as a pigment, they will appear like a blue-blooded animals, right? So here blue blood cells are available whenever oxygenated hemocyanin is available in their blood, right? So this is the way hemocyanin is imparting blue color. That is because of the presence of copper metal as a coloring pigment, right? Hemoglobin exists in the form of red color. That is because of the existence of iron metal. Iron imparting blood red color, whereas hemocyanin is imparting blue color, right? So this is the three-dimensional representation for this hemocyanin, how the hemocyanin molecules bulk level of hemocyanin molecules are appearing in three-dimensional pathway that is represented in this picture. And here you can notice exact chemical composition of that hemocyanin whenever it is oxygenated. Oxygenated hemocyanin is manifested over here. You can observe here the imidazole moieties are located, and here histidine units also located for these imidazole. And moreover, three imidazole molecules are connecting with a metal called copper, right? In the similar manner, remaining three imidazole molecules are connecting with the copper metal once again, right? So these two copper are responsible for the oxygen transportation, just like hemoglobin. How hemoglobin is transporting that oxygen throughout the organs whichever present in the body, that oxygen pumping, oxygen transportation is the essential duty of hemoglobin. The similar manner, hemocyanin is also resembling. Instead of iron, you can choose copper metal. Rather than one metallic iron, you can go with here two copper metals are accompanied by the oxygen molecule to transport, right? So this is the way hemocyanin chemical composition is manifested. This is regarding the compound called hemocyanin. Let me go with the agenda for the session. In this session, we are going to cover with the intro for this hemocyanin and what significance we are having, why we are supposed to study about this compound, the significant study we are going with, and biosynthetic strategy. What are the biological pathways are available in order to fabricate that hemocyanin within the living organisms, especially crustaceans, molluscans, and also the kind of water animals, aquatic species, and moreover the animals which are not accompanied by the bone. So backbone absent animals are supposed to accompany with this kind of hemocyanin as a coloring pigment to carry the oxygen transportation. And we are going to discuss about properties in general and chemical reactivity and potential applications regarding this hemocyanin. This is about the agenda for the session. We are going to discuss about all these concepts in detail in the session, right? A brief intro regarding the hemocyanin we are going to manifest over here, right? Hemocyanin introduction is demonstrated. Here is the snail you can clearly observe. Snail is the kind of molluscan species which is covered with the shell. It is the shelled animal. The shell is made up of calcium carbonate that is used to give the protection for the external conditions. And this is the snail which is accompanied by the coloring pigment in that blade, that is hemocyanin. Hemocyanin means for it is the copper containing protein, right? Hemocyanin is the kind of protein, and the presence of hemocyanin responsible for the transportation of the oxygen molecule in the blood for the molluscan species and arthropods. Arthropoda is a kind of huge, largest family on the earth. Largest living family on the earth will be the arthropods only in those what we can say a kind of insects. Arthropodas are simply said to be insects. In those species, again, hemocyanin is the kind of coloring pigment. Where copper existence responsible for the existence of blue color whenever that hemocyanin is oxygenated. It is binding with oxygen and giving rise to a blue coloration. This is the way hemocyanin is the kind of material as a part of bioinorganic chemistry and taking oxygen transportation and many more. Let me continue with the introduction regarding the hemocyanin. Hemocyanin is the copper containing protein and oxygen carrying protein it will be. In the blood, it is available, especially in invertebrates. Invertebrates stands for the animals which are accompanied with no backbone. Backbone absent species are said to be invertebrates, right? Such as molluscans like octopus and snails and arthropodas. Arthropodas are nothing but insects only. Crabs and spiders are the category of arthropodas. Unlike hemoglobin, which contains iron, hemoglobin is accompanied by the iron coloring pigment inducing red color over there. So that blood existing in a red color for hemoglobin due to the iron existence. Hemocyanin, there you can observe the copper metal. The copper metal is giving a blood a bluish coloration whenever it is oxygenated. This is the distinctive factor among the hemoglobin and hemocyanin, right? So both are coloring pigments only, but iron matters for the red coloration. Copper matters for the blue coloration for that blood, right? So here is the kind of a complexation between the hemocyanin with oxygen whenever we are supposed to participate in a respiration process. Those species of invertebrates. Invertebrate stands for the animals which are not accompanied by the backbone are said to be invertebrate species, right? Vertebrates are nothing but backbone supported, skeleton supported species are called vertebrates. Human is the better example for the vertebrate species, and molluscans, crustaceans, arthropods, and any other species, especially aquatic. Mostly they are aquatic in nature. Those species are accompanied by this hemocyanin, copper containing what blood is available over there. Here you can clearly observe imidazole, which is connected with the histidine unit, and here is the again another imidazole with histidine. Here imidazole with histidine. Three imidazole nucleus are accompanied by the histidine units over there, and another copper is also. It is the kind of dimer form we can say, each fragment is accompanied by one copper and three imidazoles and three histidines once again. And same will be replicated one more time. This is said to be a kind of dimer form whenever oxygenated, right? And another meta form will be, this is said to be dimer form and meta form will be there. In that meta form, what happens? There is a kind of units, whatever imidazoles and histidines are available remains as such, and copper metals also available. Instead of two oxygens, one oxygen is forming a bridge among the two copper metals. This is said to be metaform. Metaform is some kind of abnormal one, not expected, not required again. And here is the oxygenated peroxy linkage, that peroxy bridging between the two copper metals is the requisite condition for that oxygen effective transportation in these invertebrate species, right? And here is the kind of deoxyform. Whenever deoxyform will be there, oxygen absence conditions are providing individual monomers. Oxygen bridging species is missing. That's the reason why two individual coppers are existing in the form of monopositive, and the essential heterocyclics like histidine units are accompanied around the copper. Three histidine units accompanied around the individual copper ion. This is the way deoxygenated form, metaoxygenated form, and dioxygenated form in the form of peroxy bridging all are available for the kind reference. This is the way hemocyanin intro is provided. Let me jump into the next segment called significance of hemocyanin. What importance we can avail from this hemocyanin coloring pigment. Let's see. significant of hemocyanin. Hemocyanin plays a crucial role in our physiology, especially in invertebrate species, particularly in molluscans and arthropods. They are highlighting that transportation of the oxygen in those species are available. Invertebrates are accompanied by the hemocyanin as the coloring pigment. Here is the starfish. You can observe butterfly will be there, spider, lobster, earthworm. Sea cucumber will be there, resembling the cucumber. That's the reason why it is manifested as a sea cucumber. Jellyfish will be there, and scorpion, octopus. All these are the categorized as invertebrate species, and these species are accompanied by the blue coloration whenever oxygenation of this hemocyanin, copper responsible for the blue color, right? Invertebrates are the animals which lack of backbone and include many phyla and species. So many species are the part of this kind of invertebrates, but the kind of condition will be lack of backbone. Backbone absence is the compulsory condition for these invertebrate species. Examples under this category are provided, and they are accompanied by the blue blood in their circulation system. Here you can observe the animals which are reflecting blue coloration because of its pigment copper, right? So these are the kind of animals which are in specific aquatic in nature. They are reptiles. They are the kind of crustaceans. They are the kind of molluscans. They are accompanied by the hemocyanin as the coloring pigment because they are inducing blue color. And the category of the animals which are exhibiting that blue coloration of the blood, which are accompanied by the hemocyanin as the coloring pigment, are manifested over there. Snails are the examples, spiders, squid is the aquatic species, Antarctic octopus is the kind of aquatic species accompanied by this hemocyanin. Horseshoe crab is also the kind of aquatic species which existing blue color blood over there. Pillbug will be there, and scorpion, lobster, prawns. Prawns are also the category of this hemocyanin, and crayfish will be there, lobsters, scallops, and barnacles. All these, when you are supposed to extract the blood from these animals, you can clearly observe the blood is existing in a blue color because of the transition metal copper. Copper is inducing this blue coloration, sky blue coloration. Here is the representation, pictorial representation for blue-blooded animals. All these are the category of the hemocyanin pigment, right? Crayfish is provided over here. Snail is the example in this category, and the spider is also the part of arthropoda, and this is accompanied by the hemocyanin. Squid is aquatic species where you can observe the copper metal over there, and scallops are the kind of scallop stands for where you are having the kind of shell over there. They are also the kind of snail related species, molluscans only. Calcium carbonate shell will be there, and Antarctic octopus you can clearly observe over here, and horseshoe crab. Horseshoe crab is also there, which is accompanied by the hemocyanin. Pillbug will be there. This is a kind of a small insect which is accompanied by the copper pigmentation, so that blue coloration of the blood is available. Here you can locate barnacles. Barnacles also the kind of tiny organisms which are the part of that aquatic species, and a scorpion. Scorpion is the kind of venom containing species in its tail. That is arthropodon species where the blue coloration of the blood because of the copper metal. And here is the lobster. Lobster is the kind of edible species. It is also the kind of edible species you can clearly observe. Again, it is the kind of aquatic animal. And here is the prawn, everybody, where it is the kind of edible species once again. And this is also accompanied by this blue-blooded animal, right? So copper is the pigment, and hemocyanin is the kind of pigment available in their blood. So these many animals are the part of blue-blooded animals. Let me introduce significance of hemocyanin. The important activity will be the oxygen transportation. Hemocyanin is responsible to transport the oxygen to all the organs in those invertebrate species. Oxygen transportation accompanied by the hemocyanin, it serves a primary oxygen carrier. Oxygen transported by them and lack of hemoglobin wherever the species the hemoglobin content is not available, those species are accompanied by the coloring agent called a hemocyanin. There the primary oxygen transportation is available by means of this hemocyanin coloring agent. The efficiently transports oxygen in hemolymph. Hemolymph stands for hemolymph is nothing but it is the kind of fluid. It is the kind of circulatory system only. In invertebrates, the blood is represented as a hemolymph. So the blood is available in the vertebrates, right? In the human body, the circulatory fluid will be the blood only. Instead, in invertebrate species, we can synonymously manifest the name as a hemolymph. It is manifested as a hemolymph, especially in a cold and low oxygen environments like deep seas. Whenever you are going and beneath of that oceans and the seas, there the oxygen content will be very less and the penetration of the sunlight will be minimal. At that condition, at deepest shallows, you can observe the kind of species where the invertebrate species are surviving at that deepest oceans and sea regions, and the hemolymph is the kind of circulatory fluid available, and they are accompanied by this hemocyanin, which is responsible for the oxygen transportation. Here is the driver, that is a hemocyanin, you can clearly observe. And oxygen is a kind of traveling species which is taken to its destination by this hemocyanin in those invertebrate species. Invertebrate species is also manifested over here. This is a very simplified diagrammatic representation is given for your interesting observation. And here the significance of the hemocyanin to be continued adoption to the environment, right? So environmental adjustment adopted by means of the animals, which are invertebrate species, unlike the hemoglobin, which operates efficiently at the higher temperature. But the species who are staying at these low temperature, low oxygenated abnormal conditions, there the hemocyanin to be adopting the kind of requisite conditions, right? So not only higher temperature always, they are also surviving at extremely lower temperature, extremely low oxygenated conditions, extremely watered regions are available. That's the reason why hemocyanin is taking adoptions in order to survive in the abnormal environment conditions as well. Even low temperatures are suitable for them and varying oxygen availability. Though oxygen content is not sufficiently available, they are surviving. They are efficiently transporting and making the organisms effective for their survival. This is the way the hemocyanin plays a crucial role. It is optimizing itself according to the surrounding conditions. This is the way environmental adaptations are the very crucial in order to effective oxygen transportation in invertebrate species by means of hemocyanin, right? This makes it ideal for the marine animals like octopus and crabs, which lives are very cold region and oxygen poor habitats. They are surviving. Like marine species are not sufficiently accompanied by the oxygen concentration, and they are not at optimum temperature conditions as well, so that low temperature and oxygen minimal oxygen availability are the abnormal conditions they are suffering with. Still they are effectively surviving because of this kind of adaptations which are manifested by this hemocyanin. It is effectively transporting, though the little amount of oxygen is there, that will be efficiently shared among all the kind of organs. And in continuation to the significance of hemocyanin, immune response is a very essential condition. Once again, immune response stands for whatever external organs or organisms are attacking external organisms whichever attacking on these and these tiny organisms, tiny invertebrate species that used to provide the defense system, develop the defense system. So immunity matters for the effective survival. That's the reason why immune response is developed by means of hemocyanin. Some studies have shown that hemocyanin plays a role in an immune response by acting as an antimicrobial protein. Antimicrobial protein will be there that used to fight against the disease causing organism. That's the reason why immune response will be elevated by means of existence of hemocyanin, right? It can enhance the immunity by binding to the pathogens and promoting phagocytosis, right? So here pathogen activity will be minimized, and that destroying the activity of the pathogens are possible and immunity will be uplifted. This is the reason why hemocyanin not only transporting the oxygen but also fighting against the diseases, right? Microbes will be effectively destroyed by means of hemocyanin. This is the way immunity response will be elevated. And biomedical and biotechnology applications also the kind of very effective application part of this very efficient significance of this hemocyanin. Hemocyanins are used as a carriers in the vaccines and immunotherapies due to their large and complex structures and immunogenic properties, right? So they obviously are exhibiting immunogenetic activity. That is the reason why they are accompanying the kind of sensitive vaccines in order to eradicate the diseases. Lifelong, the kind of vaccines will be injected into the kind of different age groups, different phases of the life. Hemocyanin is developing immunity protection within the organisms, and also kind of carrier for various vaccines. It is used to work like a platform in order to transport the kind of vaccines, drugs, and potential disease curative agent. That's the reason why that biomedical applications are very pivotal for these hemocyanin bioinorganic compounds. They're also used in biomarkers. Biomarkers stands for they are working like a indicators. They are working like a devices which used to give something information regarding the health condition and environment as well, right? So that's the reason why they are said to be informative objects. They are so that we can treat them as a biomarkers for environmental changes. Whenever environment is changing certain extent, immediately they will give the signals, they will give the notifications regarding the kind of what changes we are about to expect, we are about to face. That kind of notification is given by the kind of hemocyanin, so that we can designate the molecule hemocyanin as a biomarker. They are giving the signs, they are giving the signals, right? But studying the protein structure and evaluation, right? Not only the environmental conditions, but also the protein structure and the kind of complexity, whatever complex biomolecules are available, the kind of detailed information is provided by these biomarkers called hemocyanin pigments, which ever taken as the dimeric form of this histidine units containing copper binding with the oxygen bridging agent. This is the reason why there is a significance in the field of biomedical and biotechnology as well. Let me go with the comparative physiology and evaluation, how the research is evolving in terms of hemocyanin applicability, right? So here, the researchers are very curious regarding the exploration of the new things. In that pathway, hemocyanin is taken as the fetching agent, being it is a kind of multi-dimensional in the applicability plethora. That's the reason why the researchers are very thirsty about this evaluation for their adaptability in various regions, especially in the physiology and evaluation process as well. That's the reason why there is a vigorous research taking upon this hemocyanin and its related analogs again. This kind of different species have evolved a unique solution for the oxygen transportation. How effective oxygen transportation is accomplished by this hemocyanin, and what way physiological applications are available, and how it is taking the part of evaluation, right? So these researchers are working in the domain of exploration for hemocyanin, where you are observing a kind of invertebrate species. Invertebrates are lacking with the backbone, and they're the kind of coordination between all the organs within the system, within the tiny system, so that how the effective transportation of oxygen is possible within the system is manifested over here. And by this, we ended up with this significant purview regarding this hemocyanin. Let me introduce that hemocyanin history. When the concept of hemocyanin began, let me enter into the history of hemocyanin. Hemocyanin was first discovered in octopus. Octopus is the kind of species in which distinctive coloration of the blood was observed, and this is said to be Octopus vulgaris, and the Leon Frederick is the scientist. Leon Frederick is the scientist who introduced the concept of this kind of hemocyanin in the year 1878. This is the way history of this hemocyanin began in the year 1878 by the eminent scientist Leon Frederick. The presence of copper in molluscans was detected even earlier by Bartolomeo Bizio in the year 1833. Entire hemocyanin existence is given by the Leo Frederick in the year 1878, but the concept of copper as a transition metal inducing the blue coloration, that concept was introduced even earlier by the eminent scientist Bartolomeo Bizio in the year 1833. This is the way the inception of the hemocyanin concept was taken in the year 1833. Copper metal responsible for the blue coloration introduced in the 1833. The entire chemical component hemocyanin introduced in the year 1878. Hemocyanins are found in the mollusca. Molluscans are accompanied by hemocyanin and even arthropod species. Cephalopods and crustaceans, and utilized by land arthropods such as tarantula, Euripalma californium, the emperor scorpion, and the centipede scutigera, Coleoprata. Also, larval storage proteins and many insects appear to be derived from the hemocyanin, right? These many species are accompanied by the kind of the pigment called hemocyanin, especially molluscans, arthropods, and the cephalopods and crustaceans are taking the part of this hemocyanin pigment coloration. A blue coloration is available. You can clearly observe this is the part of octopus. The eminent scientist Leon Frederick invention in the year 1878 was done by this eminent scientist called Leon Frederick. And this is the symbol for the history of this hemocyanin. And all these together regarded as molluscans, those who are covered with the shell, hard shell, which is fabricated by the calcium carbonate, just like the eggshell. Here also we are observing a kind of shell, snails. These shells are made by the calcium carbonate to give the external protection, and these many arthropods are available in the nature, in the universe again, and they're also taking part of this hemocyanin pigmentation, and these are said to be crustaceans, and here you can locate the crustacean species. This is the way hemocyanin is history was began in the year 1878 and even early 1833. Now, let me discuss regarding the biosynthetic plethora of the hemocyanin, how the biosynthesis of hemocyanin was accomplished, right? So, hemocyanin biosynthesis was taken in the multiple steps. So many complicated steps are required to be accomplished in order to synthesize this hemocyanin in the living organisms in invertebrate species. Hemocyanin is not a simple molecule, rather it is the complex multi-subunit protein and synthesized through the series of processes in organism cells, right? instead of the individual within the single step, rather we have to accompany by the multiple steps in order to synthesize this hemocyanin within the living organisms, right? So biosynthesis of hemocyanin primarily occurs in the specialized cells, which are called hepatopancreatic cells. Hepatopancreatic cells are nothing but liver and pancreatic cells together are taking, they are taking the platform for the biosynthesis of hemocyanin, and these cells are responsible for the hemocyanin synthesis in molluscan species, shell containing species. And hemocytes are either spots where the synthesis of this hemocyanin is available, and these are present in arthropod species, right? So here, hepatopancreatic cells in molluscans, you can clearly observe these are the origin. These are the starting point for the synthesis of this hemocyanin. Here is also the kind of hepatopancreatic cells you can locate in various organisms, right? Here is the crab, here are the molluscan species, and arthropod species also provided where hemocytes are the kind of organs where the hemocyanin manufacturing is possible. This is where biosynthesis will be originated in either hepatopancreatic cells or else hemocytes. That depends upon the type of type of organism you are choosing. If molluscans, so that the fabrication is accompanied hepatopancreatic cells, and arthropodans, hemocytes are responsible for hemocyanin biosynthetic pathway. Here are certain steps that are available for this hemocyanin biosynthesis. Gene transcription is the starting step. Gene transcription stands for hemocyanin gene is transcribed in the nucleus to produce mRNA. mRNA stands for messenger RNA, right? Transcription is the process whenever DNA is accompanied by the messenger RNA, they will interact together and giving rise to a new dimension of that a new genetic code will be revealed that is said to be a process of transcription. DNA transcription is a crucial biological process, involves the synthesis of RNA molecules which are starting with the DNA template. We are taking the double helical structure of the DNA from which new RNA species will be originated by blending with the messenger RNA, so that new kind of binding procedure will be accomplished, giving rise to RNA synthesis. This is the way DNA transcription is possible. The extent of transcription, the extent of the process of transcription accompanied by this hemocyanin biosynthetic pathway is manifested by this this is the diagnostic tool, which is immunoprecipitated in vitro translation products, polypeptide synthesized in vitro translation of the Selenese hepatopancreatic mRNA were precipitated with the anti-hemocyanin antibodies immobilized on cephalose 4B. Unbound translation products were washed away, and the antibody bound products will be electrophorosed, visualized by the autoradiography. The molecular weight of the most intense band will be 73. So this is the way the blot paper analysis will be used in order to observe how the transcription process is accomplished in these cells. Which cells? We are taking hepatopancreatic cells where mRNA is going to assist this DNA, giving rise to synthesis of RNA is possible, which is observed on this blot paper analysis. And in continuation to that one, transcription followed with the translation process, and the protein folding is also possible. Here is the ribosome. Ribosome is the kind of organ where the kind of translation process is possible. Amino acid sequence is taken part of that one, and tRNA is located upon this ribosome. tRNA stands for transfer RNA, and mRNA stands for messenger RNA, and ribosomes small subunits are available. Here is the big unit you can locate in the upper region. Ribosome large subunit will be there, and at the bottom region you can locate the smaller subunit. These are accomplished by the various number of codons over there, and here is the terminal messenger RNA. Here is the transfer RNA are taken part of that one, and the sequence of amino acids are also the part of this ribosome. Translation stands for it is the process associated with the translator, translation process required with the translator. This is a molecule translates the substrate where the mRNA is translated into a new protein. Messenger RNA is taking part with this DNA. They are going to be blended in such a manner. We are taking the strand of this DNA, and this is the strand, the red color is the strand of mRNA. Both are supposed to be blended in such a manner, giving rise to a new protein. This is the process said to be translation. The translation process involves the reading the genetic code in the mRNA to make the proteins, right? So it is also the part of protein synthesis only. Translation requires the genetic code, observation of the genetic code upon the mRNA. You can clearly observe mRNA is taken part over there. And this mRNA genetic code sequence will be observed, analyzed, and giving rise to a new protein. That new protein synthesis taking by means of this translation process in the device called a ribosome. And again, blood paper analysis is used to observe how extent this kind of biosynthetic pathway is progressing, right? So this is also the part of hemocyanin biosynthesis. And translation protein folding are taking in another kind of organ that is said to be a kind of endoplasmic reticulum. Here, translation and the protein folding are taken on the ribosomes. Here you can clearly observe the ribosomes. Two parts of the ribosomes are there. Upper region, the lower region. Upper region is somewhat wider, and the lower region is somewhat narrow in nature. And translation occurs in the ribosomes often associated with the rough endoplasmic reticulum. This is said to be Golgi complex. And this Golgi complex, rough endoplasmic reticulum is located at the bottom. That there, the translation process will be possible, right? Um, Biosynthesis of hemocyanin, we are discussing about translation and the protein folding, right? So translation stands for where the protein synthesis is accomplished by reading that mRNA. mRNA is going to transformed into a kind of protein, and that process is said to be translation. During this translation process, protein will fold itself, right? A kind of curvation is possible within. So alpha helical structure you can notice for this protein. Protein is nothing but a kind of condensed product of amino acid. Various amino acids are available. These amino acids are supposed to be condensed, giving rise to multiple number of proteins within the living organisms. Proteins are very essential and healthy survival, and they are said to be building blocks, right? This is the way they are supposed to be folded in such a manner. Helical structure is appeared. That three-dimensional representation is provided. All the kind of amino acids are taking part of that kind of helical representation, right? So this is the way kind of amino acid sequence where polypeptide chains you can locate. This is said to be amide linkage, again amide. All amide, polyamide linkages, polymeric chains are available, and moreover, a kind of curvature is due to the existence of hydrogen bonding among hydrogen bonding about to twist the molecule in such a manner, giving rise to a protein folding, right? So the primary polypeptide chain undergoes the folding and modification, glycosylation, right? So glycosylation stands for after getting that kind of twisting of the molecule that's supposed to bind up with the sugar moieties. Sugar moieties are going to attach. Proteins are supposed to be attached with the sugar molecule. That process is said to be glycosylation, which stabilizes the protein and enhances its functionality. Whatever stability parameters are required, those stability parameters will be elevated and its functionality will be enhanced. This is the reason why biosynthesis of hemocyanin, where the protein folding and followed by glycosylation are also the steps involved in this biosynthetic pathway. Let me incorporate this copper. Copper metal incorporation is requisite step over there. Whatever histidine chains we are fabricated, now we are supposed to insert the copper metal in the form of copper plus one, right? Copper existing in two different oxidation states. One is a monopositive and other is the dipositive form, right? Whenever deoxygenated hemocyanin is there, that existing in a monopositive form, whenever it is binding with the oxygen carrying with the dipositive charge. That's the reason why this hemocyanin is said to be oxygen carrier and able to transform the plus 1 to plus 2 and plus 2 to plus 1 vice versa. Copper is incorporated into the protein at a specific binding sites. Wherever histidine molecules are located, they are supposed to. Histidines work like ligands. They are forming the coordinate covalent bond with the copper metals. That's the reason why copper insertion is accomplished in between the histidine ligands. And moreover, two monomeric forms are required in order to combine with the oxygen in the form of peroxy bridging bridging form, so that it is said to be a dimeric one, right? So dimeric form will be originated in between the two copper metal. This is the reason why copper metal insertion is accomplished in between the protein whichever synthesized, so that able to dimerize with the oxygen, giving rise to a peroxy oxygen bridging. Copper is incorporated into the protein at the specific binding site coordinated by the histidine residues. Histidines are said to be amino acids which are transformed into a protein. Copper ions are typically obtained. The dietary sources are transported to the hemocyanin producing the cells, right? Whichever taken by the food, whichever dietary sources are available from that copper metal is taken inside the organism, so that that copper is the origin for this hemocyanin copper incorporation. So this is the way copper insertion is accomplished in between the histidine units, and the coordinate covalent complexation is possible, and that further dimerized with the oxygen, giving rise to oxygenated hemocyanin. Here is the three-dimensional representation is provided, where bond indicates all the bonds are inclined towards the viewer, and the dotted line bond indicated away from the viewer, and the normal line bonds indicates the moieties are located or upon the plane. This is the way copper incorporation is accomplished, so that hemocyanin biosynthesis is accomplished. Now, quaternary structure is a fabricated. Quaternary structure is nothing but hemocyanin usually exists in a multimeric form. It is not a monomer. It is not a dimer. Rather, it is the kind of multimer. Multimer is said to be more number of units. Such hemocyanin units are gathering together, giving rise to a huge one is said to be multimer. Multiple subunits are forming hexamer or else dodecamer structure, right? So here hexamer stands for 6 units are located, and deca is nothing but 10. Dodeca stands for 12 monomeric units. If taking part of that kind of huge molecule, hemocyanin will be a kind of multimer. It will be oligomer in the regular sense. We can say hemocyanin is either connected with the 6 number of units said to be hexamer. Dodeca is said to be 12 units are taking part of that multimer polymer structure. The subunits are assembled into a cylindrical or the spherical configuration, forming two or more hexameric rings. It is the kind of a cylindrical form representation, and 3D representation is also provided. Internal structure is manifested over here for the hemocyanin arrangement. This is the way cylindrical or the spherical representation is provided whenever all the units are supposed to be compacted in either hexameric or dodecameric form. This is the way quaternary structure assembly is manifested for this hemocyanin biosynthetic pathway. And in this biosynthesis separation, the functional activity will be provided Mature hemocyanin is secreted into the hemolymph. Hemolymph is nothing but a liquid whichever, just resembling the blood in the vertebrate species. In the human body, entire circulatory fluid is said to be a blood, right? So in that maximum extent, water will be there, and neither plasma cells and blood cells will be there. That exact resemblance you can observe in the invertebrate species as a hemolymph. That hemolymph in which the secretion of the hemocyanin is possible, where the circulation taking place, that hemocyanin in contact with this hemolymph, that is circulated in the entire organs of those invertebrate species and binding with the oxygen. That is the requisite function for this hemocyanin. Once in the hemolymph, it adopts its active oxygen carrying configuration. Whenever it is secreted into the hemolymph, now it is active and high affinity towards the binding with oxygen and giving rise to circulatory pigment, that circulation of the oxygen in the entire metabolic regions. Wherever metabolism is required, there the secretion and the transportation of this oxygen is possible, so that it is efficient oxygen carrier in the invertebrate species. Hemolymph is the fluid that circulates the interior arthropods and some molluscan bodies, containing the hemocytes, proteins, sugars, lipids, and hemocyanin as well, right? So that is the kind of circulatory system, circulatory fluid, especially in the arthropods and even molluscans as well. And within this hemolymph, you can observe the kind of components. One is said to be hemocyte, a kind of cells which is accompanied by the kind of circulatory fluid, and the proteins also taken part of that composition. Sugars are also located over there. Protein must bind up with the sugar so that glycolysis is possible, and lipids are nothing but fats only. They're also the part of that hemolymph composition, and hemocyanin is the pigment which imparting the blue coloration and binding with the oxygen, giving rise to oxygen transportation. This is the way a kind of secretion and followed by the transportation is accomplished by this hemocyanin. Now, let me enter into the structural dimensions of the hemocyanin, how its structure is exhibited. Hemocyanin is said to be metalloprotein, where the copper is the metal responsible for this coordinate covalent complex, giving rise to a kind of dimeric form whenever it is oxygenated. These metalloproteins contains the two copper units rather than iron that reversibly bind with the single oxygen molecule. One oxygen molecule bind with the two units of this hemocyanin, and the two copper are binding with this oxygen in a bridging manner. Oligomeric with a monomer containing a pair of copper atoms close proximity. Oligomeric in nature. Oligomer stands for the units are reaching up to 20, right? So less than 20 units if present, that is said to be oligomeric in nature. Either hexamer or dodecamer is the kind of existence for this hemocyanin. That's the reason why we can categorize this hemocyanin as a oligomeric in nature. Hemocyanins are isolated from the arthropods and molluscans and hexameric in nature, while those from the molluscans are 10 to 20 subunits are located, right? So whenever mollusca is the kind of organism you are experimenting, there hemocyanin existing in a hexameric in nature, 6 units are polymerized, right? Whereas another species, so arthropodas, if you are considering arthropodas and molluscans are hexameric in nature, and some other molluscans may possess 10 to 20 units also. Instead of 6, the number may exceeds that unit and may reaches up to 10 to 20 units are also possible, so that hemocyanin is the kind of oligomeric in nature. Deoxyhemocyanin contains the two copper unit per subunit and colorless. Whenever oxygen is not present, oxygen absent conditions are colorless only. There is no color. Whenever they are binding with the oxygen, so that the intense blue color will be appeared. That is because of this complexation, bridging complexation is imparting that blue coloration, where copper plus 1 is turned into copper plus 2. if you observe the entire structural functionality of the hemocyanin, each monomer contains the two cuprous units. Cuprous stands for copper plus one oxidation state is available that existing in monomeric form, and that able to that having strong tendency to bind up with the dioxygen. An empty cavity is present between. So there is a cavity between these units, which is sufficient for the incorporation of the oxygen. So that insertion of oxygen taking in between these two copper 2 units, so that bridging of oxygen is possible in between because of this cavity. Copper plus one is designated as a cuprous, and that bond distance is 460 picometers. No direct interaction between them. If you observe the distance between the two copper which are adjacent side by side, the distance is measured as the 460 picometers. Coordination number of the copper plus one is 3, and is satisfied by 3 histidine residues from which are taken part of the protein, right? So coordination number is 3 only. 3 is nothing but copper is able to form 3 bonds with 3 histidine units. Here is the histidine, here is the histidine, here is. That is the reason why 3 possible bonds existing around this copper plus 1 unit, and another copper is also accompanied by 3 histidine units as well. That's the reason why both the coppers are 3 coordinated in nature, deoxy form, right? This results in the distorted trigonal pyramidal geometry. Whenever 3 bonds are located, 3 bonds are not in the static, rather it is a kind of distorted one. That's the reason why distorted trigonal pyramidal geometry is accompanied by this copper. That is accompanied by the trigonal pyramidal geometry. Two phenylalanine residues, which are in the close proximity to the histidine residues, provide the hydrophobic environment at the active site, right? So here phenylalanine units are present. Whenever phenylalanine units are present in the near vicinity of these histidine units, they are repelling with the water, giving rise to hydrophobic nature. Water repelling tendency will be there. Water hating tendency is provided. That is due to the presence of phenylalanine. Phenylalanine is providing the kind of water repelling tendency. This is the way hemocyanin is molecular dimensions are represented. Now, binding of dioxygen. As of now, we've gone through the individual copper tri-coordinated complexes are taken, and this is about the deoxy form of that hemocyanin only. Let me introduce this oxygen into this cavity. Wherever cavity is present in between these two tri-coordinated copper metals, that cavity is very susceptible for the insertion of the diatomic oxygen. Wherever oxygen is not there, that is existing in a colorless form. Whenever oxygen is bounded, that is in blue in color, that is manifested in this chemical composition representation as well. Whenever dioxygen was taken, dioxygen in general is a kind of diatomic, and moreover bond order is 2. That means a 2 double bond is located in between the two oxygens. That oxygen is supposed to be inserted in between the two copper metals, giving rise to a bridged one. Whenever bridged oxygen is generated, that will be shifted from molecular oxygen into the peroxide form. Peroxide stands for, or two oxygens are bonded with a single bond, and moreover, two oxygens are accompanied by individual negative charge as well. That is a peroxide linkage. Two oxygens are directly linked with a single bond. The kind of binding with the oxygen among these two copper metals are able to release the two electrons. Two electron release process it will be, right? So the binding and release of oxygen corresponds to the two electron reaction. If you observe, copper plus 1 is supposed to be binded with oxygen, giving rise to copper plus 2. The electrons, whichever individual copper able to release one electron, right, being monopositive shifted to dipositive, so that one electron per copper. So the two coppers are releasing two electrons. Those two electrons are accepted by the oxygen and giving rise to O2 plus minus 2. So that copper is oxidized. Oxygen will be reduced, so that it is a kind of redox reaction. Oxidative addition of the dioxygen occurs. Electron transfer accompanies oxygen binding, so that copper plus 1 turned into copper plus 2, or O2 0 will be shifted to O2 minus 2. Molecular oxygen turned into peroxyoxygen. Color of the protein changes from colorless to blue in color. That is because initially copper plus 1 is D10 configurated, wherever copper plus 2 is a D9 configurated. D9 configurated is responsible for the unpaired electron, which inducing the coloration for the compound. Active site structure of the oxyhemoglobin. How the oxyhemoglobin is represented? It is a geometrical representation, trigonal pyramidal. Trigonal pyramidal is a kind of copper is accompanied by the 3 histidine units in its vicinity. And the second complex as well. This is the way each copper complex is pyramidal in nature, 3 bonds and a kind of lone pair electrons as well, so that it is the distorted pyramidal trigonal pyramidal geometry accompanied by the deoxyhemocyanin. Whenever oxygen peroxy linkage is generated in between 2 copper units, so that that able to give a square pyramidal geometry. This is said to be a square pyramidal. Each copper is accompanied by 5 bonds around, so that it is called a square pyramidal geometry. Coordination number of the copper changes from 3 to 5. Initially, 3 units are there. 3 units are surrounding around the copper, and after the binding with oxygen, giving rise to 5 bonds around, so that 5 bonds are originated from the 3 bonds. So geometry will be elongated. Geometry of the copper changes from a initially trigonal pyramidal. It will be shifted into square pyramidal. This is the way transition from the geometry is possible. The equatorial planes has a 2 histidine imidazole nitrogens, and the bonded oxygen is axially coordinated. This is the way orientation is represented. To accommodate the binding oxygen, the protein edges its conformations and bringing 2 copper atoms closer together. Whichever monomeric copper complexes are available in the deoxygenated form, they are synchronizing in such a manner, and they are binding with oxygen, giving rise to a kind of symmetrical molecule after that dimerization, oxyhemocyanin formation. Oxygen binding site is formulated as a copper plus 2, oxygen minus 2, and copper plus 2 is again. Coordination of the oxygen occurs between the 2 copper atoms, giving diheptamano. Dihepta compound formation is possible, and being it is a bridge, so that it is represented with the mu symbol, mu indicator for the bridging ligand. Now, distinctive chemical composition of the oxy and the deoxyhemoglobin. Let me compare how oxyhemoglobin is somewhat variated with the deoxyhemoglobin. That variation we are supposed to discuss about. Deoxyhemoglobin is said to be a kind of monomeric form, where trigonal pyramid is the geometry accompanied, and dimeric form is a kind of square pyramidal in nature, and deoxy form is a colorless one. Oxy is the colored one. Deoxy is the D10 configuration. Oxy is the D9 configuration. Odd electrons are absent. Odd electrons are present in the oxyhemocyanin. This is the way some distinctive factors you can compare between the deoxy and the oxyhemocyanin as well. Whenever the 2 individual units are supposed to bind up with this oxygen and giving rise to oxygen bridging in between the 2 copper metal, which is said to be oxygenation of hemocyanin, which is existing in a blue color. Experimental observation, how this oxygen is existing in a peroxy linkage, what evidence you can observe for this bond order change from double bond to a single bond, how the experimental evidence is giving justice for this peroxy linkage among the 2 copper units. Evidence is taken by recording the Raman spectroscopy for this peroxy linkage. Frequency values of the oxygen stretching frequency observed at 744 centimeter inverse. 744 centimeter inverse is observed, that is responsible for the peroxy linkage within the molecule, that confirming the existence of oxygen single bond rather than the double bond existence. Lowering the bond order from 2 to 1. Bond order initially molecular oxygen is 2, but whenever peroxy linkage is generated, that is shrinking into a 1. Peroxy bond order will be 1 only. 2 atoms are bonded with only single bond. Oxygen unit is bound in the bridging mode with oxygen bond length of 140 picometers. Oxygen bond length is approximately lower, and 140 picometers is measured in between the two oxygens whichever bonded in between the two copper metals, right? That is very typical to form the peroxy complexes. Very typical linkage you can notice, that is 140 picometers only. Coordination of the dioxygen to copper is very symmetrical in nature. Both the sides we are having exactly identical units of the copper complexes. That's the reason why it is more symmetrical in nature. Copper plus 2 centers are strongly antiferromagnetically coupled. Antiferromagnetism is a characteristic for copper plus 2. And here bridging mu oxygen peroxy ligand is being involved in the super exchange mechanism. That's the reason why antiferromagnetism is a characteristic of this copper plus 2 because of the oxygen nature. And it is the 3D representation is provided whenever copper metals are binding with the dioxygen with this bridging peroxy linkage. Deoxy form is provided and oxy form as well. Both are said to be three-dimensional representations for the hemocyanin. Now let me show mechanism of the oxygen transportation by the hemocyanin. How the hemocyanin is transporting the oxygen in the internal organs of invertebrate species. It has been noted that the species using hemocyanin for this oxygen transportation includes the crustaceans living in the very cold environments with the low oxygen pressure, low oxygen availability, but the temperatures are extremely lower at that condition. At that cryogenic condition, the species whichever existing is taking part of this hemocyanin as a transporting agent for the oxygen. Under these circumstances, hemoglobin oxygen transportation is less efficient than the hemocyanin oxygen transportation. If you compare the strength, hemoglobin is higher in potential of carrying oxygen in comparison to the hemocyanin. Nevertheless, these species are purely depending upon this hemocyanin as a carrier, efficient carrier for the transportation of oxygen to various organs in the abnormal temperature and oxygen availability conditions as well. This is the way kind of monomeric units are present, copper containing histidine units are present, which are taking part of this oxygen as a bridge and giving rise to a kind of dimeric substance over there. This is the way hemocyanin oxygen transportation is manifested over there. Some hemocyanins, horseshoe crabs or some other species of arthropods, cooperative binding is observed. Hill coefficient is provided as 1.6 to 3. So here the species like horseshoe crab and also the kind of arthropods, the binding is in such a manner cooperative binding. The nature of that binding will be cooperative binding is observed over. In these cases, cooperative binding hemocyanin was arranged in the protein subcomplexes of the 6 units, hexameric units in that term. Cooperative binding is playing a role for the oxygen transportation. Crustaceans are provided over there. The crab is the part of this crustacean. Only lobster is there, and shrimp is the part of this crustacean. And hermit crab is also the kind of crustacean you can observe. All these are circulating their oxygen by means of this hemocyanin pigment. There is a distinctive comparison between these deoxyhemocyanin and oxyhemocyanin as well, how they are differing with each other. Hemocyanin is, one can see that it is a dicopper center, which is a deoxy form. Hemocyanin in the deoxy form, there are two copper metallic monopositive charges containing deoxy form is available. Once the oxygen binding in between the two coppers, that able to generate a color change. Colorless substance will be shifted to blue in color. Colorless in nature for recorded for this deoxy form and a blue coloration for the oxyhemocyanin. Deoxyhemocyanin, where the copper is accompanied by +1 charge only, but oxygenation is enhancing the charge of copper into +2. Tends to bind with the double bonded oxygen. Double bonded oxygen binding affinity is extremely larger for these deoxy form. Oxygenated, you can observe the peroxy linkage. Oxygen nature will be shifted to doubly bonded to the singly bonded peroxy linkage is observed in the oxygenated hemocyanin. Deoxyhemocyanin copper center is +1 and D10 configuration is accorded for them, and they are in general colorless and diamagnetic in nature. But oxyhemocyanin, where the copper is accompanied by the +2 oxidation state, and D9 is the configuration accorded, one of the electron lost by this oxygenation process by individual copper. Blue coloration as the consequence, and antiferromagnetic behavior is accorded for this oxyhemocyanin. Copper geometry is a trigonal pyramidal for this deoxyhemocyanin. It will be enhanced or square pyramidal. Square pyramidal is given for oxyhemocyanin. Two copper metal internuclear distance will be very longer, being both are detached, and that is approximately 744 centimeter inverse whenever non-bonding will be there. Whenever they bonded with oxygen, 140 picometer, it is shrinking. So this is the way copper metal internuclear distance is very longer whenever deoxy form is there. It will be shortened whenever they are bonded with this oxyhemoglobin, that is 360 picometers is accorded for oxygenated form. This is the way distinctive comparison is provided between the oxygenated, deoxygenated hemocyanin. Now, let me discuss regarding this application segment of hemocyanin. Hemocyanin is taking part of this immunotherapy and the vaccine development. Immunity boosting is the very important criteria by this hemocyanin in addition to the oxygen transportation. Keyhole limpet hemocyanin is the kind of material which is very commonly used for the hemocyanin medicinal research. So in order to explore the research, keyhole limpet hemocyanin is the material very choosy in nature. There the research is in the multifold in nature in order to understand applicability, extending applicability of this hemocyanin, right? Immunotherapy and vaccine development is available for them. Immunity is the common criteria provided wherever hemocyanin taken in addition to the oxygen transportation. Immunity is also boosted by the existence of hemocyanin, right? Antigens will be developed. Widely used in the cancer immunotherapy. So they that able to combat the cancer cells. So cancer immunotherapy is the consequence of this hemocyanin. As a vaccine adjuvant to boost the body's immunity response, right? So it is the vaccine carrier as well, instead of the kind of immunity development, but also the vaccine carrier. It will be able to transport the vaccines within the organs. Diagnostic and research applications of the hemocyanin, it is used as the heptene carrier in the immunological studies. As we already aware, immunity boosting is the common as a criteria which is accorded for this hemocyanin. In addition, immunogenic proteins aids generating antibodies in the research. New antibodies will be developed in the research purview by using this hemocyanin in the laboratory. It works like a biomarker, able to sense the chemical changes of the environment, and environmental sensitive changes will be detected and monitored by means of the kind of this hemocyanin, so that environmental stress, environmental degradation will be immediately monitored and alarmed by means of this hemocyanin, right? The heptane concept describes the most frequent mode of sensitization of this chemicals need to bind with the proteins, forming a heptane carrier complex. Heptane carrier complex will be originated to be able to present the particular T cell receptor as the heptanated peptide and to induce the specific immunity response. Whatever immunological system developed within the organ where the heptane carrier is taken the responsibility of this hemocyanin, that is developing immunity response. So this kind of mechanism is provided by this hemocyanin, which is oxygenated over here. In addition, antimicrobial property not only developing the immunity, rather it able to kill the pathogens as well, so that viruses can be killed, and so many microbes will be damaged and injured by means of this kind of hemocyanin, so that it is effective antibiotic. And multiple researchers are working in the segment of this applicability of this hemocyanin in the various disease curation and disease diagnosis as well. This is the way virus replication will be inhibited, and antibiotics will be developed, and the research is on progress. As we already aware, hemocyanin is the better environmental monitoring agent. It able to give the sensor sensitivity. It able to absorb the sensitivity of the environment. Slight change in the environment will be noticed by means of this hemocyanin. Hence, it is a kind of pollution biomarker. Assessment of the nature and the extent of the exposure, how extent the environment got changed, how extent it got damage, that kind of information gathered by this hemocyanin. It is the biological indicator, biomarker, we can say. Assessment of the molecular and the cellular alteration, how extent cell metabolism got modified, that also assessed by means of this hemocyanin. Assessment of susceptibility of the pollutants, how these pollutants are susceptible to induce the diseases, what impact generated by them, that impact is also assessed by means of this hemocyanin. This is the way it is the pollution biomarker agent, right? So this is hemocyanin, right? Biotechnology and the protein studies by hemocyanin, it able to give the valuable information regarding the primary structure, secondary structure, tertiary, and even quaternary structure of the protein. Protein structure will be revealed by means of this hemocyanin. Primary structure is simply said to be what a sequence of the amino acid taking part of the simple chain, and whenever this is taking the part of curvature, helical structure is induced whenever they are binding with the hydrogen bonding system, that able to give the kind of curvature within the molecule is said to be a secondary one. Whenever these secondary structures are taking part of some extent of complexity, right? So polypeptide chains are developed, and they are binding with themselves within the intramolecular bonding system, that able to give a kind of complexity within, which is said to be tertiary. And still more complexity is noticed within the quaternary structure. The aggregation of all kind of polypeptide chains are possible, giving the very huge complex web, which is said to be a quaternary. All these structural methodologies will be revealed by means of this hemocyanin, so that it is taking part of a very potential biotechnology tool, and protein structural studies will be revealed by means of hemocyanin. It serves a model protein for the studying protein structure, folding, and a copper binding mechanisms as well. And comparative biochemistry will be very valuable in the field of research platform, and how effective oxygen transportation possible among all the different species. That comparative study is also efficiently maintained by this hemocyanin pigment. Catalytic activity will be there. They works like a catalyst, and especially like a polyphenol oxidase. They works like a polyphenol oxidase. Whenever the kind of tyrosine and L-DOPA will be there, it is a neurotransmitter. Tyrosine is the amino acid taken over there. They are oxygenated by means of this hemocyanin. It works like a oxidase enzyme. Enzyme activity is also taken part of that one. That's the reason why tyrosine is monohydroxylated initially. Later, it will be inserted with the additional hydroxyl. This is the way oxidation. A polyphenolic oxidation in enzymatic activity, catalytic activity will be taken part by this hemocyanin, right? So this is the L-DOPA neurotransmitter will be shifted from hydroxyl into a ketonic one again. Removal of hydrogen is said to be a kind of oxidation. This is the way it works, like a polyphenol oxidase enzyme. Catalytic activity giving rise to a kind of corresponding products as well, right? Hemocyanin is the homologous to the phenol oxidase enzyme, and tyrokinase enzyme again. So it works like a oxygenation process, or else removal of the hydrogen, so that oxidation process will be accomplished in the presence of hemocyanin, right? This is the way hemocyanin exhibiting polyphenol oxidase activity. Let's find the difference between the hemocyanin and hemoglobin. A kind of both are having the same intention of oxygen transportation. What the difference you can notice among these two kind of biomolecules? Let me show. Right, the kind of hemoglobin is a red coloration, lead coloring pigment, where the iron taking part of the center in between the entire porphyrin ring, where pyrrole rings are a heterocyclic ring system taken, and alkyl side chains are also the kind of system located, and the conjugation is essentiality, and coordinate covalent bonds are very symmetrical in between the porphyrin ring system and the iron metal, which is dye positive in nature. This is the structural representation for the hemoglobin. If you observe hemocyanin under oxygenated form, where two copper metals are taking over there, and the histidine units are playing the part of this heterocyclic chemistry, and it is again symmetrical in nature in the oxygen taking a bridge in between the molecule and responsible for the blue coloration of the blood. If you observe the hemoglobin and hemocyanin together, metallic component taken part of hemoglobin will be the iron transition metal and hemocyanin copper transition metal. Hemoglobin is exhibiting red color, whereas hemocyanin is faintly bluish in nature, right? Hemoglobin plasma of some invertebrates. You can locate this hemoglobin, and RBC cells of all the vertebrates, the essential component will be the hemoglobin only. Hemocyanin is located within the fluids of crustaceans, that is said to be hemolymph. In the crustaceans, arthropodans and molluscans, cephalospodans, the body fluid which is said to be hemolymph, there the existence of hemocyanin is possible. Transportation of the respiratory gases, able to carry the oxygen and carbon dioxide, obviously. Whenever oxygen is given, carbon dioxide taken up. This is said to be exchange of the gases in between the organs and the kind of circulatory system. That's the reason why they're able to carry the both oxygen and carbon dioxide in the case of hemoglobin. Whereas hemocyanin, the susceptibility of the oxygen carrying capacity is manifested, but not carbon dioxide. This is the way comparative studies provided for hemoglobin and hemocyanin together. Three-dimensional representation is provided whenever three-dimensional representation is observed. The molecule is supposed to be twisted. Whenever you're rotating the molecule 90 degrees, it is manifested like this. This is the way three-dimensional form in the molluscan hemocyanin is provided. That is observed in the molluscan Japanese flying squid. Japanese flying squid, where the hemocyanin is extracted and whose three-dimensional representation is analyzed. So this is said to be cylindrical in nature, and each monomer has a string of 8 individual subunits. Copper oxide is the part of this kind of coloring pigment. This is the way the molecule is provided for the molluscan Japanese flying squid. By this, we successfully accomplished the entire session. Hopefully, this is very informative towards the scholars, researchers, and also the scientists, academicians, and organic chemistry, curious people, and those who are serious about the entrance examination and the competitive exams as well. The kind of session is very informative towards those seekers. And the session conducted by Dr. Kavita Kothireddy Kota. I'm an academician and researcher, and the entire content is available on the channel World of Competitive Chemistry. In addition, we conducted a series of sessions on the organic chemistry, heterocyclic chemistry, alkaloids and terpenoids. So many compounds are available, and even intermediate chemistry is also available over there. And for the entrance examinations, whatever you are preparing about the chemistry, the entire content you can find at the platform called the World of Competitive Chemistry. I felt you enjoyed the session, and really it is informative towards the viewer. And kindly subscribe and share with the aspirants who are more curious about the chemistry. And in the near future, we are going to conduct the series of sessions based on various concepts which are really helpful. Thank you very much for your patience.